Now, let's enter into topic 10, and this is the last topic, but first we have to go to 10.1 and then 10.2. So, we have to see first of all 10.1. It talks about the sexual reproduction and genetics. So, basically, in the sexual reproduction, we are going to talk about the second type of cell division. Cell cycle is divided into interphase, then M phase. This M phase is divided into mitosis and cytokinesis. But not always mitosis. It can be mitosis or meiosis. Now, in the sexual reproduction, we deal with meiosis, this type, okay, because meiosis is the one which produces gametes. So, first of all, as per the KPI, we need to know what are homologous chromosomes and how they look like. So, the chromosomes that make up a pair, they are always in a pair. One come from each parent. They are called as homologous chromosomes. You can see in these pictures about the homologous chromosomes, like how they look like. So if you have a look at this diagram, any of these, you can easily check that these circles are at the same place. It means that they have the same genes. Okay, and then above that, you see from here to here, both have the same height, or you can say same size. So till now we studied they have same size, then same genes. Now the most important thing, the location or the position of this centromere. So same position of centromere. But there is another thing that we have to notice. The genes that code for earlobe type, yani any type, might not code for the exact same type. So not always, like both genes may not code the same. Okay, so this is very important to understand. This is the chromosome. We did the labeling before. This is centromere. These are the sister chromatids, and then this is a pair of the homologous chromosomes. So basically, what are the homologous chromosomes? They have same genes, same size, and the same position on of the centromere. Next one, we have merged some of the KPIs 10.1.2, 1.3, 1.7. Describe the specific events that occur within each stage of meiosis 1 and meiosis 2. So it means that meiosis is divided into two substages, not like mitosis. We need to sketch them and we need to observe them, yani we need to identify them. So that's why all are related to meiosis. How we define meiosis? Meiosis is a type of cell division that reduces the number of the chromosomes. It means that reduces to half. Okay, reduces to half, always remember. That's why we also know it's known as reduction phase. So normally, if the parent cell has 24 chromosomes, if it goes to meiosis, the daughter cell will have 12 chromosomes. Okay, so it has 24 chromosomes, but this one has 12 chromosomes. Okay, further we need to identify the stages of each. So we will start from meiosis 1. Before meiosis 1, we will have interphase 1. This diagram is really very important. Why? Because it has the identification of diagrams and the reasons why we do it. So for example, if this diagram is given to you in the exam, this one, what you will name it? Definitely interphase. Why? Because the chromosomes are replicating and the chromatin started to condense. The spindle fibers are almost near the poles, and you can see the nucleus still. After that, we have prophase 1, where the pair of the homologous chromosomes come close to each other, and crossing over takes place. This is very, very important. Crossing over that takes place in prophase 1. Then the nuclear envelope breaks down, and rest is same as mitosis. So this is difference, one of the difference between mitosis and meiosis. Metaphase 1, chromosomes centromere attached to the spindle fibers, but remember that these are not normal chromosomes. These are pair of chromosomes known as the homologous chromosomes. In metaphase, you have see on the equator only one chromosome per spindle, but over here two chromosomes per spindle. And just because of that, we don't know which one will go to this pole or which one will go to this pole. So that's why we say that it follows independent assortment. Assault mean yani how they will be classified or like that. So it is independent assortment. Then we will see in the anaphase same thing, but over here you see a full chromosome moving to opposite pole. So we will say that the homologous chromosomes separate, not the centromere divide. In the telophase one you have two full cells, and then in cytokinesis, of course, it will divide into two cells. Moving to the second half, that is the prophase 2. In prophase 2 again the same process will be repeated, but over here now you have only one chromosome at the equator. Okay, but the easy hint is that you have two cells over here, so you can easily say that this is meiosis 2. Then the same, the sister chromatids will divide. Now here the centromere splits, sister chromatids go separate. Okay, they move to opposite pole, and this is all because of the centromere split. Now after that we have telophase 2. In the telophase 2 we get 1, 2, 3 and 4 cells, and cell divide, spindle fiber breakdown, and that's it. Now you will have a product that is 4 cells. You can easily see over here products, 4 cells. If you remember, the mitosis gives you 2 cells, so this is another difference. Then each nucleus contains a haploid chromosome number. This is another difference. So these are the differences between mitosis and meiosis. Tricky question that you have to identify each phase of meiosis and make sure you say it 1 or 2. So first of all, if you look at the first picture, the chromosomes are in V shape, okay, and it means that they are moving to opposite poles, but you can easily see that they are homologous chromosomes. So that's why this should be anaphase 1. Then you have like 2 cells, okay, and you don't have a full chromosome, you have just half of the chromosome. So that's why this will be again anaphase, but this time it is anaphase 2. Then you can see that the chromosomes are exchanging their segments. So this is prophase 1. It's easy to identify because the crossing over takes place in this one. And after that we have chromosomes lined up in the center, and you can see that the homologous chromosomes are lined up, so 2 2 in the center. So that's why this is metaphase 1. And then over here, as 1 1 chromosomes are lined up, and also it's 2 cells, so it is metaphase 2. And in addition to that, now you have 2 cells. You can easily see over here you are going to have 2 cells, so it is telophase 1. And in the end, now you have over here 1 2 3 4, so this is telophase 2. And over here you have 2 cells, and both are like almost rising the centromeres and spindles and everything. That's why we can say it as the cells are in prophase 2. So with that we did the labeling. Now let's move to the question. The difference between anaphase of mitosis and anaphase 1 of meiosis is that the chromosomes line up. No, lining up is metaphase. Remember, the chromosomes line up is metaphase. Centromeres do not exist in, no. Chromatids do not separate at the centromere in anaphase 1. Yes, that's true, because chromatids don't separate. the whole chromosome move from one place, from one pole to another place. And crossing over occurs in anaphase of mitosis. No, this is anaphase of not even anaphase, sorry, this should be prophase, because crossing over takes place only in the prophase, and it should be meiosis, not mitosis. Okay, moving further, we have more questions. Using the diagram information you have learned about how cell divide, you have 5 figures A, B, C, D, E. We need to check which diagram above depicts means shows, okay, or represents a cell at the end of meiosis 1. So at the end of meiosis 1, you should have 2 cells only, and each cell should have a full chromosome. Okay, so meiosis 1, so it means that it's going to be D, like the cell E. Which of the diagram above depicts the cell at the end of meiosis 2? Now we have meiosis 2. It should have 4 cells, and each cell, meiosis 2 results in 4 cells, and each cell has half chromosome number. So see, only A has the half chromosome number out of all. So next question, which of the following diagrams depicts the cell at the beginning of mitosis? So mitosis, it should have the full chromosome number at the beginning and at the end. So that's why we can easily say that the cell D is the one that has the full chromosome number. Moving to next question, which of the diagrams above depicts the cell at the end of mitosis? End of mitosis. So again, if mitosis starts with 46 chromosomes, okay, it will end up with a cell with 46 chromosomes. So once the cycle is completed, once the mitosis is completed, you get the whole chromosome number over here. So definitely it is B. Okay, let me show you. This is B. So you can see over here it started with D and then it ends up with B, like each cell will have the whole chromosome number. The chromosome shown represents a pair of the chromosomes. So what they are called, definitely we studied, they get one from parent, one from father, one from mother. So these are known as the homologous chromosomes. Next one, describe how chromatids exchange their genetic material during synapses and crossing over. What is their importance? And then there was another KPI which is 10.1.12, that is almost same. We need to talk about when the crossing over takes place and when independent assortment takes place. So basically there are 3 main processes that are very important to increase the genetic variation. One of these, so the 3 main processes, one of those is crossing over, that takes place in the prophase 1 of meiosis. Second, independent assortment, that takes place in the metaphase. And then random mating during fertilization of the sperm and egg. So we will talk about these again. Now, the most important thing over here is the crossing over, cross over. So the chromosomes, they cross their segments. So you see there are 2 homologous chromosomes, they come align, then they start to exchange a little part of it. By the way, to know that it's not always at the bottom, it can be at the top as well. Then they exchange a little part. Now you can see it was green, now this segment of green, it is like, let's say, reddish golden, so it's over here now. So they have exchange, and then they go back into their normal positions. Now they have different parts of the genes. That's why it leads to genetic variation. Crossing over occurs in, of course, prophase 1. Then the which of the processes shown over here? Of course, crossing over, no need to look into it. If they say that select 2, so you can say second is synapses. Okay, here a lot of KPIs are merged, like 10.1.8. 10.1.10, 11, and 13, and all talks about the same thing: fertilization. Meiosis results in gametes, and gametes receive half of the chromosome. They join together to make the zygote. Okay, like sexual reproduction is the transmission of genetic information from offsprings um, by the help of egg and sperm. So all concepts are same. If you understand this diagram, you will be able to understand everything, and you will be able to answer anything in the exam. So first of all, you need to see that this is male and female. Both of these, they undergo a process A, and then they result in the production of cell X. Okay, now we need to know what is cell X. This shape is very familiar and common, and the cell, and they are like this. So it means that they are gametes. Okay, so let me write in the box directly. So cell X, they are gametes. Now we need to see process A. So gametes are always produced by meiosis, so definitely this is going to be meiosis. If this is sperm and this is egg, they join together to make cell Y, that will be definitely zygote. And then process B is fertilization. So you have to identify when these gametes join together, they undergo a process of fertilization, and they result in the formation of a zygote. This zygote divide and redivide by the help of mitosis, keeps going throughout the lifespan of them, and they result in the production of adult male or adult female. So we can easily say over here that process B is fertilization, and Y is Y. Yeah, Y is what? It's a zygote. So we need to know like what are their functions. Like process A is meiosis, so its function is to produce gametes. Okay, fertilization, sperm and egg join to redevelop the 46 chromosome number. Gametes are sperms and eggs, and zygote produced by the process of fertilization under shock. And the zygote is the cell that is produced by the process of fertilization, resulting into a complete cell chromosome. What process reestablishes the diploid chromosome number? Reestablish mean build again, to make again. So like let's say sperm has 23, it joins with egg that has 23, it will re-establish the into zygote, and this zygote is going to have 46 chromosome number. Okay, so the process that re-establishes is of course fertilization. In human gamete, they are haploid. The chromosome number is dash, while the zygote are diploid and the chromosome number is dash. You know that the human have 46 chromosome number, so you can choose easily. But look at this one. The first one should be haploid. So when you look at haploid, it should be 23. Now we can easily check from here 23 and 46. Yes, this one. And animals with a diploid number of 36 will have a chromosome in its gametes. So 36 divided by 2 is equal to 18. And its body cells, of course, 36 again. So now 18 and 36, this option. Compare and contrast mitosis and meiosis, include their stages and everything and outcome. You can easily look over here, like parent cell divide into two cells. So one cell divide into two cells, but over here one cell divide into four cells. So one cell divides into four new daughter cells. Each daughter cell has half chromosome number if it is a gamete. Mitosis, one division occurs during mitosis, but over here we have two divisions, meiosis 1 and meiosis 2. DNA replication occurs during interphase. DNA replication occurs just before meiosis 1. Synapsis of the homologous chromosomes does not occur in mitosis. They do occur over here. 2 cells are produced, 4 cells are produced. The daughter cells are genetically identical. They are not genetically identical because of crossing over. Mitosis occurs only in the body cells. Yes, this is very important. And then it occurs in the reproductive cells. Mitosis is involved in growth and repair, while meiosis is involved in the production of gametes, providing genetic variation, and etc. You can easily tick over here, like if it is mitosis or meiosis. You can pause the video and just go over the answers, try them. For example, I will do one for you in both cases. Homologous chromosomes pair together. Where they pair? Definitely they paired in meiosis. Something like that, you can just go over it. Next table, it says that you have to write the correct number for each statement, like 1 2 3 4 5. So number of the DNA. So in the second question, how many number of DNA replication? So of course in mitosis we already know it is 1, but also in meiosis it is 1, and so on. Okay, chromosome number of the daughter cells is 2n and n. Number of cell divisions 1 and 2, meiosis 1, meiosis 2. Number of daughter cells 2 and 4. Next one, describe the sorry, let me tell you, we have touched 10.2. That's the last topic. Okay, so we are almost done. Discuss the contribution of Gregor Mendel to study genetics. It's all reading. There is no critical thinking behind it. This is just like discuss. So you can say that the Gregor Mendel worked on the pea plants and he discovered the fundamental laws of inheritance. These are his contributions. Like, what did he do? So Mendel deduced the genes that come in pairs. Mendel tracked the segregation of parental genes and whether it is dominant or recessive. Explain why Gregor Mendel used tube breeding. So basically, why he used tube breeding, saying that pea plants are true breed, mean that they consistently produce offsprings with only one form of the traits. So pea plants, basically they gave you only one characteristics to be studied further. That's why he limited his study to the true breedings. Differentiate between self-fertilization and cross-fertilization. The words themselves, self and cross, they show the meaning. Self mean fertilization, you remember, fusion of sperm and egg. Self mean within the same flower. So if the fusion of the gametes, male gamete and the female gamete, is taking place in the same flower, this is known as the self-fertilization. But if they go to another flower, so this will be known as cross fertilization. To prevent self fertilization, Mendel removed the male organ of the flower of the yellow seed plant, and then Mendel called the green seed plants and yellow seed plants. So it's also known as the P generation. So basically, the idea behind it, to tell you that Mendel did himself the cross fertilization. Explain why Gregor Mendel used cross pollination to study. Yes, this is the reason. The point was to test how different traits are inherited. When Mendel cross pollinated one variety of the pure breed plant with another, these crosses would yield offsprings that looked like either one of the parent or not a blend of the two. Number 5, which is the second last KPI, define the terms. Heredity. It is also known as inheritance. It is the passing of traits from one generation to the next generation. Genetics, study of heredity or study of genes. Genotype is an organism's allele pair. Phenotype is the observable characteristics that you can see. Phenotype is the physical appearance of an organism or observable characteristics of organisms. Gene is the segment of the DNA. It is the smallest segment of the DNA that is responsible to produce a trait. Allele are the alternative forms of the gene. So basically, in the figure below, you can see that these are different forms of alleles. So like the pea plant has to self fertilize or the cross fertilize. So here the pea plant, it's based on the seed color is yellow, capital Y, capital Y, it's like an allele. Then this is the second allele. So it means that each gene has minimum two alleles. Each gene has minimum two alleles. Okay, last one is done. Now we have questions. Which of the following statements are true about Gregor Mendel and his importance to biology? He is the father of genetics? Yes. He explained heredity? Yes. He explained how dominance could mask the effect of presence? Yes. So it means that all of these. Mendel used only dash traits. We know that he used pure breeds. Sometime you don't need to read the whole question. It clicks in your mind very quickly. Gregor Mendel contributed to genetics by concluding that the traits are inherited by the next generation. Okay, the traits are inherited by the next generation. Gregor Mendel is known as the father of genetics because he is the father first to conclude that traits are inherited from parents to offsprings. And the last question, no, sorry, second last. Gregor Mendel used pea plants in his study because they're the true breeding, meaning that they give rise to only one trait. We studied it already. The offsprings will only form with one trait. Last question. What process was used by Mendel? Of course, it was cross pollination because he took the male and he then male part and sprinkle on the female part. They resulted in the pollination, and then the new plants were produced with the same traits. Thank you so much. You are all amazing. Wish you all the best, and I hope that you will be preparing it well. If there is any question, please feel free to let me know. And there we go with a big thank you for the whole term that you spent, and thank you so much.